Hello, everybody. So my name is John Zhu, Department Biology, NMSU, and I'm responsible for this toxicity mice study. Okay, Yan already gave us an overall idea and what's going on. So in different levels of the models and the cell lines and the zebrafish, which is vertebrate very early and evolution-wise, and a mouse. So we're gonna use. So here is more close to humans, right? It's the mammal. It's a pretty good model system, easy to culture them, to raise them. So here we use this outbred mice and try to see, usually, so for medical research, you use very pure, so inbred. And that one we consider, say, there's a population, all different individual variations, there's a pretty big range. That's why we chose this outbred mice. On water, we have 3 different types. In the control water, it's a city water, tap water, with some activated carbon and post-carbon and plus UV. And the loaded and raw produced water. And here we have like 5% of raw produced water and just try to lower the toxicity. We don't want to kill the mice right away. And salt control in here, the difference there, so there's a high salt in raw produced water. So we use a salt control only contains 7 grams of sodium chloride. And then to treated produced water. So one is the desalinated, and the others like TDS and the TOC is relatively high level, and particularly ammonia and the treatment 2. So salt removal plus post-treatment. So now, and ammonia went down concentration. It's the best treatment, I think. Oops, supposed to be this way. Sorry. So in the mouse the groups, we have acute study, 28-day exposure. So here we have females and males, each 5 mice in there. And so for the raw produced water, so 5%, we have a 10 days exposure. That time we didn't have enough mice, and each group got 2 mice. So overall now we can see the weight gain. So here comparison only post-treatment, TW1, TW2, compared to control. And looks like the female mice grow a little bit faster in the first 2 weeks on the produced water too. It's more pure water. Looks like the mice like better. So liver body weight. Here, liver enlargement can be an indicator of like a damage or highly active activity, that kind of thing. So here we can see the raw produced water and the control, and females particularly control and have a pretty bigger liver size. And the males, very similar situation. So the salt control have the liver enlargement. So after treatment, they all look the same or very similar to the control, no significant difference. So, and here's the blood test, hematology. We have cells or different measures. And right there, we can see 5% raw produced water or salt control. Again, they show some hemoglobin concentration went down. So after treatment, they all very similar to, were identical to the control water. So here's the comparison. So salt control caused some hemoglobin change, downregulate. And right here, some other measures right there. And after treatment, TW1, which still have a pretty high ammonia concentration, the sodium went high a little bit. And now all these raw produced water or salt control and with some phosphorus, so up. And globulin went down, the female, and produced water. And treatment 2, pretty good, everything fine. Right. So histology here, we only compared, we only have the slides for tissue sections for post-treatment, 2 different waters. So overall, they look very, very similar to the control. We don't see anything very different. So now, it's a major part. So we know there's a lot response. So if you expose to some toxins, so here, toxicity, and it can cause damage and repairing and injury, and also, so C, the toxicity, they're gonna have the defense mechanisms all respond to the different conditions. It's very complicated. So, and liver transcriptome, because we know liver is the major tissue or for the toxicity defense. And liver also is very important and produce a lot of proteins and for all kind of different activities, right? So that's why we check the liver transcriptome. So why transcriptome? Because now in biology say we need a lot of proteins to play all kind of cellular activities. So enzymes, structure proteins, so and so. But to characterize proteins, not easy. But now we know, so you need a protein, then you need to have a template to make proteins. What's the template then? It's a so-called RNA, messenger RNA. So if we can measure messenger RNA, then we can guess, so what's gonna happen at a protein level, right? So that's a very easy, technology available to do that. So advantage is that we don't need to know anything before we do this. So no prior knowledge required, and which is unbiased, right? We just see what's going on. So, but disadvantage right there is say, you have a damage, you have a repairing system response, and a detoxification, they all come back. So, and make this data analysis sometimes difficult. Okay, so we're in that process. So here, the RNA sampling, so the males and the females, each, we took 3 the mice to do this because the cost, the budget, and exposure for the raw produced water. So we have only have 2 mice, we use them all. So that's the sampling. So here, totally. So in the mouse genome, so there's a 40,000 and 400 predicted protein coding genes. And from this entire data set, so in 11,000 genes detectable. So with different expression level, and here we use a so-called TPM to represent expression level. Here we can see, so it's 1 to 10, it's relatively low expression level. Here we have 57% genes in that level, in the middle abundance. So here is less than 4,000. And the high abundant genes, these are close to 1,000. Okay. So, and we validate, say, the RNA-seq data, how reliable usually you have to use the individual gene, so qPCR to validate. So all these 4 things for the female mosquitoes, sorry, I'm working on mosquitoes, I always say mosquitoes, mice, all right? So, and the response is very similar pattern like RNA-seq. So here, the males, we have 5 genes, for validation purpose, they all go the same pattern like RNA-seq. So validate, so that we pretty much, RNA-seq is pretty much reliable. So, and because now we know there's 11,000 genes, how can we do it individually? No, almost impossible. So luckily, so there's a methodology analysis tool called PCA. So try to organize all these 11,000 genes expression pattern in the different conditions, then we can make it a little bit easier to see overall. So first, let's see. here, you see a line right there. Above, you can see that's the response pattern organized together. It's in response to the salt. So in the males right there on the top, 2 purple dots represent 2 mice. So they go there, and the females, and it's all above there. But sorry. So the males, purple, and the females, kind of like blue, so and separated. That means the sex show the different response. And compared to the raw produced water and versus control is on this side. Also separate male from female. So that's a pretty interesting pattern. So and also data analysis summarize them together so we can see this clear pattern. Sex difference, also the salt versus raw produced water, they are different. Oh, it's more importantly here, you see close to 60% variance across 11,000 genes, different expression pattern. So we can see actually sort them out, right? So they summarize about 60% variance there. It's a very effective method. Right there, so now we can put them all together, the females, look at here. So salt and the raw produce, the other side, now the controls and the 2 post-treatment produced water, TW1, TW2, they go together to the normal control.
Sorry, on the previous slide, can you clarify what you mean by components, those 3 components that control that 59% of the variance?
Okay, PC 1, 2, 3, that's what you're talking about?
Just the statement says 3 components. What is a component? Is it one?
Oh, okay. So that's a PC1, PC2, PC3. So in the 3-dimensional, right, okay, that's the methodology and the PCA, okay. Sorry, I didn't explain that. So right here, now you see, so the raw, after treatment and the control, they all go together this side, and the salt or raw produced water the other side. So here's the female data, and now the male data the same way. So above there, I'm sorry. So salt and raw produced water here now, and after treatment is very close to the tap water control. Same pattern. So then we have a different way to look at it. Genes upregulated. That means more in response to the treatment. So here, how are we going to rate this? This one is like a statistic support. The higher means the p-value is extremely small. And right here is the so-called upregulation compared to the control, this side. Here is the downregulation means abundance lower of the treatment. So that's this side. So the lines defines 4 different quadrants. Here, 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 and here. Below lines, the gray dots means all these genes are not significantly different, or the fold change is lower, less than 2-fold. It's more relatively stringent. So right here, you can see the raw produced water versus control. That's the tap water control. And here we can see 80 different genes. They show the differential expression pattern. Here, 33 downregulated, 47 upregulated. But after treatment in the PW12, so with the salt removal, so the 11 remained, 10 remained. So that means, and removed 73%, just the salt removal. So this side, the TW2, the treatment 2, also removed some other stuff like ammonia. So only 2 remained. How many together right there? So 80, so the other side, okay. So it's relatively efficient, removal all these effectors. So here, the male data, 137 differentially expressed genes, downregulated, upregulated, but TW1 treatment, So 7 remain, 7 remain. The other side is 8 and 16 remain. So here we try to include it more like an even lower expression level, like a TPM 10. Okay. So overall, now we can see the treatment remove a lot of factors. So put them all together, and we can see, so 11,000 genes detectable, and we used PCA to sort out all the data. We can see the transcriptome response is a sex differentiated, and the raw produced water induce a lot of genes, and upregulated or downregulated, but treatment to remove most of them. So, and here where we have ongoing subchronic, which is going to be 90 days exposure, right? So it's still ongoing, and we don't have anything to show today. Then overall, the mouse toxicity study, you can see, so the raw produced water and the control cause some in histology or biochemical changes, and so they can represent some like damage response, repairing response, or toxicity, like detoxification type. So, and here, the treatment and pretty effectively remove some effectors. And I think that's all I have here. Yeah, so, and the funding from this consortium, and here's the biomedical research facility this afternoon. We're gonna have a tour there. It's a brand new building. We are 2 of the labs using this facility. Very useful. Okay, so that's the team. All right, so, and that's all I have, and yeah, okay.